basically, I mentioned earlier about the National Tissue Bank, which basically started with the help of Professor Andy Hall up in Newcastle. But he always said from the outset, I can get this going, but I can't be the permanent home. We can't be the permanent home for your bank. So we've been very fortunate to, through our process of trying to collect tissue and get it transported to various places, etc., so that it can be used in research, to have made contact with Janine Salter, who runs the tissue bank at the Royal Marsden. And Janine very kindly offered to take over the bank from Newcastle. And as I mentioned earlier, it's been a rather convoluted process to get there, but we're quite close now. So I'd like to hand you over to Janine so she can talk about GIST biobanking and why it's really important to research.
Okay, thank you. Good morning, everyone. I'd actually like to start this with a quick caveat that I'm not a clinician and I'm not a GIST expert. So I think from that basis, what I'm going to tell you is much more about a generic way of looking at things, or looking at biobanking. My background is breast cancer research. I've been worked for breast cancer research for 18, almost 20 years. And in that time, has been a very important part of that research, which is translational, is looking at collecting materials for researchers so that they can actually look into all these various mutations and different problems that we see with cancer. What I'm going to do today, I'm going to take you back a bit. You've had a lot of very sort of specific science here about all your different mutations and the effects. I'm going to take you back a bit, back to sort of the beginning of all of this, so I can sort of get you on a journey with me to show you that what we're trying to do is an interaction between basic science, translational science, and then through to the patient treatment. So I'm going to take you on that journey, if you bear with me. Some of you will probably find this a bit simplistic, but I just want to get you with me so you understand why we're trying to do what we're doing. So what happens? I want to start with a normal situation. So what's happening in your body normally? Your body turns over cells continuously. If you're perfectly normal, you have no disease whatsoever, your normal processes are that your cells will renew. And in order to do that, they have to listen to cues from outside through what we call receptors, or either on the cell surface or in the nucleus itself, to tell it to do certain things. And it also has another part of its process in the cells. It always has in its sort of genetic makeup the ability to do the opposite, which is actually to stop proliferating and growing and actually die. That process, what they call programmed cell death, is called apoptosis. And I noticed in one of Ian's slides, it's up there as one of the features of the disease. So what we're having, we're getting cues from around the body, various things in the body, talking to the cells, saying, come on, mate, keep multiplying, keep doing what you're doing, I need you to do your job. Through the process of that multiplication, or through the process of its work, those cells start to get a little bit tired, and they start to find problems with the way they're functioning, etc., etc. So through that process, it then gets to the point where the body then says, hang on a minute, you're not doing your job properly anymore, you need to now die. And it starts sending a different set of signals to the cell. The cell listens to that and goes, okay, that's fine, now I'm going to stop growing and I'm actually going to take myself down a programmed cell death route. That's great when it works. What happens in the cancer cell, which is quite a different thing entirely, the cancer cell itself starts ignoring those, oh, mate, you've got to die messages, and just keeps listening to the ones that tells it to keep proliferating. So effectively is what you've got in my cartoon here is your cell is sitting there. The signals are still coming, but the cell's just going, I'm a teenager, I'm not listening to you. I'm not going to listen to any of those messages you're sending me. I'm going to listen to the other ones. It's going to keep me going. I'm going to keep doing the things I want to do. And that's what happens in a cancer cell, and you get this uncontrolled survival and proliferation. And that's what you're seeing in your cancer. So what does this all mean, and why am I telling you all this? Well, in order to actually do a cycle of what I call a cycle of research, you have to go through a number of processes to get to the point of killing the cancer or wiping the cancer out. And in order to do that, we have various levels. We have the basic research, which is done by the scientists locked away, no one ever sees. most of the time. You get the translational stuff, which is the stuff that actually moves into more relevant to the patient. And then you've got the clinical trials, which Ian's spoken a lot about, the various trials that you have ongoing to actually test those hypotheses actually in the patient, make sure it actually does what it says it's going to do. Okay, so that's sort of the main sort of process and what you would expect as you go through these. And obviously you've got the various in the clinical setting, which you'll obviously all be very aware of, the various phases of trials. You know, the phase 1, which is all looking about whether this will actually work, and then later on you get higher and bigger and wider cohorts of patients to actually see that you're actually getting some benefit. So what's making up that research portfolio I'm talking about? So on the top level here, most of this is what you're talking about in terms of basic research. So you're looking at what Ian was talking about, blood cell growth. That's angiogenesis. That's the posh name for it. But basically what it's doing is recruiting, teaching it how to keep pulling blood vessels into the cancer, because cancer needs that to survive. So it's very clever. The cancer cell's very clever. It will do all these things to actually make it work. Avoid apoptosis, which is the cell death. Pull in blood vessels to make it grow faster and better because it gets all the nutrients it needs. Cell proliferation, it differentiates, does different things than it used to do, because now it's got this other survival instinct. Then the bottom half is when we start looking at the translational stuff, which is looking at the sort of targets, the gene therapies. They're actually now starting to look at the effect it has in the body. Cancer genetics is probably one of the biggest growing areas at the moment, and those of you who know about the 100,000 Genome Project, is a real big thing about looking at the genes and what's actually causing a lot of the problems in cancer and rare disease for that program. So, what are the challenges then? So, what we're trying to do is to move from that basic science, the crazy one in the laboratory, doing all the great stuff with seeing what actually happens in a cell and actually relating it to nothing else. And then we've got to make that transition from that into the actual patient, the patient material. You know, what's the use of knowing about what this particular receptor does and what it does in that cell and what it kicks out at the end? You know, you've got to have a reason for doing that in cancer. So that's where our translational bit comes in. We've got a big gap there. And we've got a big gap there because what you need in order to do that translation in the first instance is patient material. That's the tumour material. The tumour material that you're holding, getting removed on a regular basis if you have cancer. That material is extremely valuable to the researcher, because in order to be able to see that what they're saying as a hypothesis in cell lines outside of the body actually will work in the context of in the real world, is I've called it there. There's also a problem when we're doing anything like this. As you all are painfully aware, GIST is quite what we call a rare disease. And unfortunately, when we're dealing with this kind of work, where we're actually trying to look at whether something works, we're caught up in the whole statistical problem of it. Is it a real event, or is it just by chance? And that, unfortunately, drives most of science, and that makes it quite difficult. So it's really important that if we can get this material that is from patients, and lots of it, it gives us more opportunity to do what we want to do and actually have the statistical power to actually do what we want. Now, we've seen this done really, and I can vouch that because I've seen it, I've watched programs march on in breast cancer and in prostate cancer, but they have the numbers. They have huge numbers of patients coming through, so they're able to do this and turn it around much quicker because of that. In the case of GIST, obviously, we're very rare. We're not going to get so many. So we really, really have to get every single patient sample we can in order to get the power to do this. This is why this is really, really important. Okay, a little bit about GIST. As I said, at the moment, as we've been told, the GIST Biobank isn't currently with the Royal Marsden yet. We're in the process of trying to move it down from Newcastle at the moment. But you already know that it's been collecting samples from patients with GIST, and they've got an access committee, and all the right things are in place. It's got ethical approval, which means they can release the samples quite easily. All that stuff's been done brilliantly and very well by Andy Hall, who you've been hearing about. So that's all in place already. I say it's in Newcastle. It's moving down to the Royal Marsden. This is the building it will be housed in, which is our Centre for Molecular Pathology down in our Sutton branch in the Marsden. This is a fantastic innovation of a building, because you have a combination of people who are working as researchers from the Institute of Cancer Research, and they're working with the clinical people as well. So what you're doing, you'll be able to see the actual science actually moving into the actual patient care. Ian spoke to you about actually monitoring patient progress through. So he was talking about actually circulating DNA, and we have a really big feature on circulating DNA in our research programs. It's key to actually not only just monitoring, but it's also maybe a way of being able to identify these diseases much earlier on, which gives us a much better chance of being able to treat them appropriately. As I say, we do need a lot of material, so this is sort of one of the reasons we want to collect as much as we can. The idea of the bank is that we want to work with be collaborative. Because of the nature of the GIST tumour, it's very rare, and we need to collaborate as far and wide as we possibly can, both nationally and internationally, to bring all these samples together. So, as I say, we have that power to make those decisions that we want to make. So it comes back to you guys. What can you do to help? Well, I think the main thing is that if you are, or you know someone who's going to have an operation for GIST, let us know. Get involved. Consent to actually storing your samples for future research. It's the only way we can do it. We can't take the material unless you actually approve of us to do it. It's one of the key features of the Human Tissue Act. And if you can talk to other patients that you meet along the way and say, well, look, this is what we're trying to do, and we really can make a difference if we all pull it together. Talk to your clinical team. Some clinicians are not actually aware that we are actually collecting GIST samples, and if you get them involved and they know about it, they're more likely to want to do it, particularly the surgeons. It's about that. And the whole point, what we're trying to do is make sure we've got those logistical things in place that we can get your sample from wherever it is around the country and bring it back to the Marsden for storage. Raising awareness, and we talk about this all the time, raising awareness to everyone. Make sure everyone knows what we're doing so you can get people involved. Let's get the message out there. And I think the most important thing, GIST is rare, but every patient is important. There's a big thing nowadays about, Ian's talked a lot about resistance, and personalised medicine is really all about trying to make sure we can tailor the drugs to your individual type of GIST. And it is different with different people. You may have the same phenotype, but it behaves differently. And there's a whole plethora of reasons why that is. And the research around is all about trying to find out what's causing that, what makes that happen. And the only way we can do that is we've got that material to try and make that come together. I love this. This is just a picture of I caught off the internet, which was a fantastic one. It's a liquid nitrogen bank, this, with all the gas coming out of it and somebody all dressed up. We wouldn't be allowed to do that in the UK. This is an Australian picture. But I just thought I'd show it because I just loved it. I thought it was lovely. But thank you. So if there's any questions, do speak.
We have a question. Thank you. Both you and Professor Judson have mentioned the difficulty that your GIST is so rare. And this is a question really directed at you, Professor, which is that is there more to be learned, because of that, is there more to be learned from the likes of treatment for CML, leukemia? I understand that Gleevec hopped over from the treatment of leukemia to GIST. Are there other things that we can learn from treatments of other conditions?
Sorry, I think this is still... Okay, I'll use this one. To answer the first question about what we can learn from CML, the idea of the alternating treatment does actually stem from experience in CML. Also, to a certain extent, the idea of using the blood, because in CML, it's of course very easy to know what's happening with the cells because the cells are in the blood. So you can isolate the leukemia cells, and you can also immediately test whether a resistant mutation's occurred and what it is. Curiously, what they found is that if you come off a drug like imatinib or nilotinib and substitute one of the old-fashioned treatments like chlorambucil, that's CLL. It's the other one they use. Anyway, an old-fashioned drug. You can see that resistant mutation, when you take away the pressure that's creating it, disappears. And so I think some of the current thinking about treatment has evolved because of what we're learning from CML. But the target protein is different. The mechanisms of resistance are different. And there isn't quite the same, I'm not an expert in leukemia, but I don't think there's quite the same spectrum of almost different diseases that there is with GIST. But yes, we have adopted some of the strategies.
Thank you, Jane.
Another question.
Jane, you asked first.
One thing I just wanted Janine hopefully to reinforce, it's very, when you're preparing for an operation, and you might talk to your local hospital about you'd like to donate your tissue to the tissue bank, because we're not kind of known nationally. Most surgeons are very busy. They might have heard of us. They would have forgotten about us. Most hospitals do also have a local bank where they store tissue. So it's very easy for them to give you a consent form for you to have tissue collected for research, but it will be collected probably in formalin rather than fresh and held in the local bank. And it won't actually then get to the central bank, which is where we're trying to stimulate the research from. So if any of you should be preparing at any point for an operation, in the first instance, till we get the bank in place at the Marsden, if you could contact me, I will make sure that things work so that your tissue does go to the tissue bank. And it needs to be fresh and frozen rather than put in formalin, which is standard for operations.
Yeah, that's right.
Thank you, Jane. I want to pick up on what you've just said. I'm completely sold on the value of the bank, and I have been from the time I first joined the GIST group. So that when the time came for my own surgery, I was quite keen that I wanted to go along with this. But it didn't happen for a very simple reason, and that was I was completely unclear on the procedure to follow. It didn't help that this was early this year, so we were again still then in the transition phase, and Jane warned me about that. I did contact Jane. I was given all kinds of paperwork to get signed, but looking back on it with hindsight, it is all left too late because I arrived at the hospital. I did not set it up in advance. I arrived at the hospital. I was seen by one of the surgeons who was going to be involved in the team, who in fact was, I suppose, inevitably a junior surgeon, who was very confused about what this was all about. And she kind of said, well, I don't really know. I'll have to speak to the lead surgeon. Keep your paperwork and we'll get it off you later. Well, I left the hospital 8 days later and the paperwork was still in my bag. So it just didn't happen. Yeah. I think, therefore, it's clear to me that if we're going to go this route, it's terribly important to get it set up well in advance. And my question then is, who do we set it up with? The oncologist, who was my oncologist, is extremely helpful and supportive. I had thought of speaking to the surgeon, but when I saw the surgeon, surgeons are not necessarily very talkative. And when I went in to see my surgeon, I was completely gobsmacked. I went in and he kind of said, well, hello, this is what we're going to do, goodbye. I was in with him for about 10 minutes. I didn't get a chance to ask nearly the questions I wanted to, and I certainly didn't get to talk about the tissue bank.
From memory, your operation, while we talked about it, us engaging to actually get things moving happened far too close to the operation. Generally, obviously, I've been assisting with the collection of fresh tissue for our bank for a couple of years now. And ideally, we need 3 weeks' notice, because the process that I go through is obviously the patient signs the consent forms, which are issued by the tissue bank. They take a copy of that to their surgeon. And then once they've given me permission to do that, I then engage with their surgeon to explain what the bank is and what the patient has asked us to try and facilitate. I also then engage with the pathology department in that hospital, because I have to find a pathologist who's willing to receive fresh tissue, select the appropriate parts of the tumour that are for the bank. We need to organise for a blood sample also to accompany that. And then having got all of that into place, which is no mean feat, because usually I don't know who these people are. I've yet to have a repeat situation where somebody's having an operation in the hospital where I've already done this once. It's then a matter of coordinating with the specialist couriers that we have to make sure they're arriving at the hospital when the operation has happened, when the tissue's been sent internally to the pathology department, and the pathology department are ready to release it. Now, operations sometimes get delayed or postponed, so that obviously is a complication. None of these things are impossible, but if we're only told about it the night before, a couple of days before, it is quite a tall order, because getting hold of surgeons to actually discuss it with them, it's not easy because they're in operations most of the time. Usually you have to track down their secretary and sort of in triplicate send them a letter and say, can they put it under their nose when they're rushing from one thing to another. So time is of the essence. So generally, just operations, if they're not emergency ones, are planned in advance. So it's just a matter of, getting things in place so that we know. I wouldn't expect you as a patient to have to do a great deal. Once you've consented and you've given that information to your surgeon and your oncologist, and I would then expect to take over that role of coordinating what happens, but time is of the essence, really.
Yeah, no, I think it's sort of mirror that really, but I think the main thing for us is that if you can let us know really as soon as possible where you're interested in and where you're like to have your operation. We have a lot of connections across the country with the various pathology departments. Now, some of them may not be willing to do this, and it would be a shame to have gone through all this effort and then we not be able to do it. But it is about us actually interacting with the pathology department in particular, because that's where the sample's coming from. Most of them are geared up now. There's a very big drive generally to store frozen material for various reasons. And I think we just have to be really clear and early. I think what Jane is saying, earlier the better, because that's when it does take a long time to set things up and convince people that it's a good idea. And it just is a shame if we miss it. And as I've said so many times, these are so precious, these samples, and if we can't get them, it would be a great shame. But we certainly will try really, really hard. But I say, sooner you can let us know, the better. And then we'll put wheels in motion.
One thing I would say at the moment, while we're in this sort of transition phase, what we've been doing, because we have had patients who've alerted us and tissue's been collected, and we can still do it. We just can't transport it to the Marsden at the moment. But what we've been asking is, are the local pathology department able to store it in the right conditions for us until we're able to then transfer it? And so far, everyone's been keen to do that.